So today I wanted to cover a little bit of what we're trying to do more on the therapeutic development side for PPP2R5D and some of the different approaches that we're taking, kind of the direction that we're going. As Jan mentioned before, my group does a lot of delivery. We kind of divided the lab very basically into delivery and cargo. And so today's a cargo talk, not a delivery talk, but happy to share what we're doing in terms of accessing the brain and delivering these gene-modifying domains and different things to neurons. If you guys are interested, some other time, I'm sure I have the talks on my laptop. I also wanted to give a quick update on where we are with the iPSCs. I didn't put the slides in there because, again, it's showing similar pictures. We have pluripotent stem cells derived from the fibroblasts from the kids from the Houston family event. 3 lines are developed. They've been characterized to be pluripotent. They're karyotypically stable. We're in the process now of converting them to a feeder-free solution or feeder-free culture conditions instead of growing them on MEFs. They're just a little bit easier for researchers to work with. That way, we do have them grown on the feeder cells too and banked. The 4th line is we have colonies, iPSC colonies, and we're in the process of characterizing them for potency and karyotypic stability. And starting in August, we have all the constructs to do the creation of the isogenic controls. So in this time while the cells have been being created, we've been working out the protocols in terms of doing the nuclear affection to be able to plate them to do what we call SIB selection. And so Rich told you earlier today about the HEC293s, which are very amenable to editing, that are very amenable to growing a single cell, that are pretty straightforward to create these clones. iPSCs are the opposite of that. So there's a lot of protocols that we've been optimizing and working out to be able to create the isogenic controls. That work's starting next week, and hopefully by the next time we'll have some preliminary data of showing what we're doing with that work. Just as an update. So the other part of my lab working on actually being able to do targeted regulation and correction of the variant lines. And so we work a lot with CRISPR-Cas9 as well as other DNA binding domains and modifying agents. And so just again, as kind of the general layout in the summary slide, there is a variant in the DNA sequence that leads to a variant RNA transcript that eventually gets translated into the variant protein, and that protein we've heard a lot about today by the biochemists in the group, of the different binding partners and the different ways that this plays a role with the different binding domains. And so some of the first things that we've been working on is just using an approach called CRISPR interference. This is very straightforward. We've done this for other disease indications in our group. And this is being able to repress the DNA or repress transcription or block gene expression at the DNA level, and that's recruiting something with a guide RNA and a Cas9 protein with these transcriptional repressors, things like CRAB, which are already abundant in mammalian cells, to regulate gene transcription. And we've shown before that we have guide RNAs designed for the promoter of PPP2R5D, and that we can repress this. The downside to such an approach, because this has to target regulatory regions of the gene itself, and so it's not allele specific. So we see a biallelic suppression of PPP2R5D, which all of the data so far suggests that that's probably not a viable approach for the kids in terms of we need the normal healthy copy. Some of the other things that we kind of decided to take a shot on goal with, that our lab does a lot of work or has done a lot of work in the past, is creating things that interfere with RNA transcription. And so these are things like short interfering RNAs. And what this does is we can design these siRNAs to try to be specific to only the variant coding into our variant transcript. And what happens if these bind, they form a complementary strand, they recruit the RISC complex, and it degrades the mRNA variant so the protein doesn't get made. These are pretty easy to make and deliver. However, the specificity of them can be highly specific, and they can also be promiscuous. And so this is kind of an empirical question of whether or not we could get these to recognize only the mutant allele and spare the wild type allele. And so we designed a lot of these constructs. Unfortunately, for the variants that we're looking at here, the way in which they were designed, we're not able to recognize that single base pair difference between our variant and our wild type allele. And so the other thing that we're really excited about moving forward in terms of therapeutics are things called RNA editors. And so these are, again, kind of new classes of using the DNA binding domains, but instead of having them recognize genomic sequences, they recognize the RNA or the transcript here. And these are the Cas13s. There's Cas13b. There's now Cas13 or Cas-Rx. And what these do is they recruit these ADARs, and these ADARs are bringing in a base, and what this does is it changes it to an adenosine that then gets converted as an I during the read and then to a G. And so in the majority of our E to K variants, what this would do is at the RNA level would take that variant RNA and switch it to the wild type to be read as the wild type protein. And so this, again, in theory, would create two copies of the wild type protein without doing irreversible changes to the genome, which we can talk about why that is sometimes scary to go in and edit and do base editing within the genome. Rich kind of mentioned it, that in certain cases you can be highly specific. In a lot of cases you get a lot of promiscuity. So more letters than the one that you're aiming at. So as I mentioned before, Cas13b is kind of the pioneering factor that was first described to do this. Cas-Rx is the more specific kind of next generation of these binding domains. And then a paper just got published last week, which we're really excited about, called LEAPer. And I have a slide at the very end. If we have time, I can show you why we're excited about what that approach could mean for Jordan syndrome. And so just a little bit of the preliminary data that we've generated. I think I've shown some of these before. Just using the CRISPR-I approaches in wild-type fibroblasts, or the fibroblasts that we've gotten from the patients, we can show that we can relatively regulate transcription within these cells. Our siRNA-mediated knockdown, what we've been able to show, and I think I showed this in one of the virtual lab meetings or in person, is that we can see knockdown of 2R5D. We also see dysregulation when we target 2R5D, that these other subunits may be doing either compensatory mechanisms, or we're seeing changes of regulation of these other subunits when we're reducing 2R5D. I don't have in here today the allele specific where we looked at this, but you'll just have to trust me that it was not allele specific, meaning that we hit both the wild type and the mutant copy. We're going through the chemistry and the design of the siRNAs now to understand whether or not if we can do further modifications to try to increase that specificity. But it may just be with the siRNA that they're just, they create these tiny hairpins over where the variant is, and so the binding kinetics are a little bit altered. Our first approach is with using Cas13b, and so we have all of our guide RNAs, in which we're just showing that in our patient-derived fibroblasts with the E198K variant, we can transfect these cells. Again, fibroblasts are notoriously hard to get things into. And what we are just showing here is that the expression levels are unchanged overall, meaning that we're not creating a premature stop codon, we're not altering the mRNA. And we were hoping to have the data late last night, but unfortunately it didn't come in, where we're looking at the allele-specific changes here to understand whether or not we're creating a skewing of, we would want to stay at 1x expression, but if we look at the wild type and the mutant in an untreated cell, they would also be at 1x, and then if our Cas13b approaches or Cas13 or Cas-Rx are working, or these other ADAR recruiting complexes, is that we'd get a skewing of more wild type allele on the read with just using regular qPCR. So these are pretty interesting for us. We're moving these forward within the patient cell lines, and then we'll start in the iPSCs when they're available, as well as in the NPCs that we'll be creating. So this is our team. Jasmine Carter, you'll get familiar with her. We'll start bringing her to the meetings. She's going to be our graduate student that is really heading a lot of this work and developing these new technologies, working with the cells. So with that, I can take questions. I can skip to Leaper because it's kind of cool. I'm excited about it. Yeah? Okay. So, all cells pending a few very specific cell types actually have mechanisms. And so one of the things that our lab really likes to do is leverage existing cell regulatory systems that our body already has and making them do other things that maybe they're not used to doing. And so this is really exciting for us is because what this does is it actually, instead of putting in, so anybody that's done anything or followed the CRISPR field or Cas9, these are bacterial proteins that our bodies and most of us probably have some levels of adaptive immunity to. And so overexpressing Cas9 or this Cas protein could cause an immune response. It could not be, there's going to be some clinical hurdles in terms of how we do it. What's exciting about this is instead of having to recruit these foreign proteins, most of our cells actually have expression of endogenous ADAR, and this ADAR is an RNA editor that goes in, like we said, that causes a single base pair of the transcript to switch. This is used in a lot of our interleukins to change gene expression somewhat naturally, and then you can recruit these with just like a guide RNA again, so something again that's going to look very natural and endogenous to our systems. You just design these different guide sequences to overlap the base or the transcript that you would like to change, and it causes, you can see here the editing events within the windows that you can see. So white means that there was no editing. Each box is the single base pair of the transcript, and that they're showing pretty high efficiencies into the 40 to 50% of being able to go in using endogenous RNA editors to switch a transcript from variant to healthy. And so they did a lot with GFP early in the paper. And one of the things that I thought was the most exciting is they actually went into primary fibroblasts from another syndrome called Hurler, which is a lysosomal storage, and are actually able to restore functional enzyme within these cells at about 30 to 40%. So we ordered all the constructs, and we're testing these now. So this paper just came out. I thought that it was pretty relevant to us as a group as something that we can try to push forward as a therapeutic angle to alter, to make the bad copy look like a good copy and to have it read as a good copy to produce a functional protein instead of figuring out other ways to do it. And then this, again, is a lot smaller of a system, is a lot more endogenous of a system, and so a lot of the delivery hurdles that we typically run into, we likely wouldn't with this. So, happy to take any questions.
And logistically, you said you're still in seeing that pretty cool. Logistically, you would make those smaller RNAs inside the cell, and then it recruits the ADAR itself.
Yeah, so ADAR. ADAR, so there's three variants of ADAR that are almost in all different in all cell types. ADAR1 seems to be the most efficient at doing RNA editing, and it's also the most abundant. It's almost ubiquitously expressed across all mammalian cell types, and in neurons. So that was the first thing that we checked outside of this paper. And then, so basically, you're creating these, in essence, longer guide RNAs. Which you could deliver with AAV or whatever. You just have to have a RNA promoter like a U6 or something. So relatively easy to package. Yeah?
Don't the ADARs just target adenosine? Convert this to cytosine? You couldn't target other nucleotides then?
So there's other endogenous ADARs that you can do, and then if you target like the antisense or the sense strand of it, sometimes you can try to get corrective events. Yeah. Any other questions? Yes.
Are you limited with 801, 2, and 3 with certain PAM, or are there?
It's not PAM restricted. So because this doesn't recruit a Cas, you don't have to be close to an NGG or any other of the canonical PAM sites. It's only where the complementary. So it's literally complementary guide binding. And so, and then instead of having like your tRNA to recruit to bind to Cas9, it's a different tag. And I think I cut it out of the figures when I was making this. But it's just something else that is basically, it recruits endogenous ADARs to that site, which is not restricted to a GG. Yeah, so it could be relatively genome-wide. It would just come down to the specificity of, the specificity of your, oh, okay. It just come down to the recognition of that guide sequence. And so again, the longer, and so you can see a lot of them are like these 113 nucleotides. And so you get relatively high transcriptional specificity because they're so long. Yeah.
So Kyle, is this you started out with 2 voice delivery or cargo?
This is cargo.
Okay. So this is a different thing to deliver.
This is something that would go in and if I go, this would work at, so again, when we think about targets outside of the small molecule drug world, something that would be going to the variant itself and doing a corrective event. There's ways, and Rich has alluded to it before, where we can work at the genomic level with base editors or doing different types of targeted things. The only problem is it's not efficient in vivo, especially in neurons. So then like antibodies or other small molecule drugs will modify protein, and what this would do would work, it's going to work here. And so it's not changing the genome, but it would change this and cause this from being read as variant to having it be read as healthy. So instead of having a healthy copy and a variant copy, it would read as 2 healthy copies and produce healthy proteins, in theory.